Well, greetings everybody from California. This is Phil Roberts, and I'm going to share with you our project area of interest 1.1, titled Development of Marker-Driven Improved Cowpea Varieties for West Africa Using Mature Markers. I wanted to first share the geographic focus of West Africa. We're working in the countries of Burkina Faso, Ghana, and Senegal, and introduce our collaborative team here. Along with myself, we have Bo Lam Huynh and Tim Close, co-PIs here at University of California, Riverside. In Burkina at Inara, we have Joseph Badiano. We have Francis Kusi in Ghana at Sari, Tamale. And then in Senegal at Isra and also Seras, we have Musa Dianga and his senior colleague Diaga Cisse. And I think everyone pretty much on the list here is participating in the global convening. So just a couple of background considerations, which I think anyone working in cowpea is very aware of. We have a highly nutritious legume, vital to food security. Real challenge is that the yields of smallholder farmers in the Sudan of Sahel are only typically about up to 20% of the known yield potential. The good news is we have downstream breeding technologies and tools that will allow us to advance cowpea quality and yield. And the project provides an excellent framework for host country capacity building, both in terms of degree training and ongoing scientist short-term training. We have 3 objectives in the project. These are sort of our global objectives. Objective 1, applying marker systems to add traits to elite African varieties. And I'll go over in some detail then on these. Objective 2 is taking lines that we've already bred through other projects and looking at their field performance in terms of yield and their quality and acceptance and nutrient value. And then third, using our marker technology tools for quality assurance of current and new varieties, seed stock purity, and so on. So let me touch first on some key points of progress on Objective 1, where we're using MABC, which is marker-assisted backcrossing. And in this program, as listed here, our team has selected 3 elite varieties, current varieties in each of the host countries. So they're doing pretty well already. And to those, using MABC to add in value-added traits, as it were, in terms of yield and grain quality. So you can see here a lot of emphasis on larger seed size in the different programs. In one case, breeding for earliness in Ghana. And then these other traits are biotic resistance targeted traits to insect, macrophymener, soil fungus, and then parasitic weed striga, pest problems where we have marker tagged resistance traits that we can move in. For insects, aphids are important in Burkina and Ghana certainly, and then flower thrips is a focus in Senegal. So how are we progressing on this? It's a stepwise process. Early in the project, we made the first F1 crosses. So this is where we cross the elite variety, what we call the recurrent parent, with trait donor parents, so where the traits are that we're going to bring in. And then we back cross into the recurrent, so we have first generation back cross. We completed this process in 2020. In the last year, under this central panel, our focus has been on advancing this first generation back cross material. And so we repeatedly back cross, and then at each back cross generation, we use the marker technology to select plants that have the best background content, so most like the recurrent parent background, and then also foreground selection for the specific traits. So it's an iterative process, and we have got out to BC2 F2 generation with work completed in Ghana and Burkina. We've had some technical difficulty with the Senegal materials, so we're a bit behind on those, but nevertheless, the process then is to come forward with each of these generations, and currently in the most advanced materials, we have, with the second backcross generation, started intercrosses to stack the different traits into these elite lines. And then going out in 2022, we will continue this iterative process to develop lines that have fixed in them all the favorable alleles for the QTL target traits. Excuse me. Just to visualize some of this work, here's a greenhouse at Riverside where we are growing out early generation back cross materials produced and then sent from Ghana, used for collecting leaf tissue for genotyping. And just to sort of emphasize here, we grow out in this panel on A, we grow out a high intensity of population, so lots of different plants. And then the marker information through genotyping allows us to eliminate most of the plants, and then we keep the ones that have the high background and trait content for the next cycle. And just to digress briefly, we've used this MABC process successfully to benefit U.S. agriculture under the program. And this has culminated in release of a new black eye variety, a dry grain variety, California Black Eye 77. I reported on this last year. This is where we took the standard grown CB46 variety and added aphid resistance donated from an African line. So this is a great example of a sort of a win-win in terms of the USAID funded US Africa relationship. And in the last year since we talked, we have this registered in the Journal of Plant Registration, so a formal release that we hope will do very well. And I just wanted to enlarge the seed image here to show that besides the added aphid resistance, we also have a slightly whiter, brighter grain type in the new variety here on the right that we think will be most attractive to consumers compared to the current standard variety. So under Objective 2, I'd like to go very briefly through excuse me, highlights of what we're doing here. And this is where we're taking existing elite breeding materials or lines and then using field-based assessment in each of the host countries to determine which of the lines are appropriate for release as new varieties. And it also, these materials are also part of the cross-cutting nutrition project, which I'll touch on at the end here. So we have lines developed previously from marker-assisted recurrent selection, and then multi-parent advanced generation population materials, including some that we've previously intercrossed that we call super magic lines. So we have a set of 69 lines that we started off with. We developed breeder seeds at UCR and then have been collecting data on yield and grain quality, pest and disease response, and so on in the host country trials. In 2021, we had 4 location grow outs across the 3 host countries, and these will then also tie in with the opportunity for training through farmer participatory variety selection. On the right, we have photos from this last summer main season showing grow outs of these elite materials used in our trials. And I just wanted to emphasize here, we have some of our degree training students providing presentations. I just give an example of 2 from Ghana here, Yor Nasamu and Samuel Owusu, who you'll be hearing from very shortly. Just to re-emphasize, we feel the project has value certainly in terms of training and capacity building through 2 avenues. One is the lab and field-based project allows training in molecular breeding, both for on a continuing basis the NARO staff, so the scientists and their lab staff, and then also the degree-based student training in the 3 host countries. And then in terms of the farmer training, the field trials are being used for participatory variety selection. So our stakeholders in the host countries are tied in. Okay, I'd like to just finish just for another minute or 2 with the cross-cutting theme project we have focused on cowpea nutrition. And as I said before, this ties in with the objective 2 elite line materials that we've been testing. And we've taken a set of these. We have selected 30 lines where we have completed now in 2021 a grain nutrient status analysis, where we're looking at sugars, protein, and some of the micronutrients. We produced grain samples in 2021. Earlier, these were used for the nutrient analyses, and then more recent samples will be used for 2022 sensory evaluation, taste evaluations, and cooking time tests. And this will be followed up with an education program on cowpea nutrition benefits. On the right is a magic line grown out in 2020 with particularly attractive grain. So one of the materials we're looking at. Again, just to emphasize, the grain samples are being produced, or have been produced in the 3 host countries, and these have serviced the nutrient content assays and then set us up for the sensory evaluations. And I'll just finish here with a summary of the nutrient content analysis in our set of 30 elite materials and 6 local varieties. And the point I wanted to bring out here is that we have an excellent range from minimum to maximum content in each of the nutrients that we're interested in. So proteins, 23% to over 30% grain content. Good ranges for, this is parts per million for calcium, iron, and zinc across the series of materials. And then sugars ranging from nearly 8% to over 13%. So we feel that the set of materials we have sets us up really well for a very useful sensory evaluation. So with that, I'll finish, and I really appreciate your time, and hopefully we have time for 1 or 2 questions. Thank you very much.
I'm presenting on preliminary phenotyping of advanced cowpea genotypes to select promising lines with multiple traits. Cowpea is a good source of dietary protein, cash, and fodder for livestock. Cowpea is drought and heat tolerant. It has a high yield potential, but the yields on farmers' field is low due to biotic factors such as pests and diseases, and abiotic factors ranging from salinity down to poor soil fertility. These factors has been the focus of research. So genotypes that withstand these factors have been developed or identified. That notwithstanding, farmers still record lower yields because they do not have access to these research materials. The objective of this study was to evaluate advanced breeding lines to identify superior agronomic performance under the Sudan Savannah zone of Ghana. The specific objectives were to determine the variability in the agronomic and yield characteristics of the cowpea genotypes, and to identify the potential assertions for further evaluation towards the release of new varieties. Materials and methods. The experiment was carried out at CSIR of Sari using 71 cowpea genotypes, out of which 3, namely Wankai, Apabala, and Kekausbenga, were the Czech genotypes, whilst the others were advanced breeding lines that had been selected for traits ranging from attractiveness to high yields. The trial was a randomized complete block design with 4 replications. Each plot was a single row of 2 meters, and seeds were planted at a planting distance of 20 centimeters between hills. Qualitative and quantitative data sets were collected based on crop descriptors. These data sets were analyzed using R. Results and discussions. On the field, 4 flower color types were identified: the purple flower, the white flower, the white with the purple margins, the white with the V-shaped purple pigmentation at the center. 49 of the accessions had white flowers, whilst 4 had purple flowers. Flower color is a predeterminant of seed coat color. In an experiment where seed coat color is the objective, flower color could be used to indirectly select for the seed coat color. Varying seed coat color and pattern were identified on the field. 59 of the accessions were either white or cream, 7 of them were mottled, 4 of them were solid brown, and these were from the purple flowered plants, and 1 was black and white. Pod position. 38 of the accessions had their pod above the canopy, whilst 26 had their pods within the canopy. Pods above the canopy are easy to harvest, either mechanically or using manual means. Pods above the canopy are also easy to monitor for pest and disease damage and to control them than when the pods are within the canopy. Table 2 shows the summary statistics of measured agronomic traits. Flowering of the cowpea genotypes was between 34 days to 52 days and averaged at 37 days for the earliest flowering genotype, whilst maturity ranged from 60 days to 82 days, averaging at 66 days. Grain yield, on the other hand, ranged from 865 to 3,349 kilograms per hectare and averaged at 1,991 kilograms per hectare. Principal component analysis showed the contribution scores and correlation of the agronomic traits to the principal components. Days to flowering and days to maturity were positively correlated with the first principal component, while grain yield and biomass yield were negatively correlated with the first principal component. Plant height was correlated with the fourth principal component, whilst 100 seed weight was also correlated with the second principal component. The distribution of the genotypes and the traits measured on the principal component by plot are presented in Figure 5. The cluster analysis further grouped the genotypes into 6 clusters. Cluster 1 was made up of the high-yielding genotypes, with an average yield of 2,818 kilograms per hectare. Cluster 2, on the other hand, was the early maturing cowpea genotypes, flowering at 35 days after planting and maturing as early as 64 days after planting. Cluster 3 and Cluster 6 were within the early maturing group. Likewise, Cluster 5, all flowering within the range of 35 to 37 days after planting and maturing within a maximum of 67 days after planting. Cluster 4, however, made up of only one genotype, was the late maturing cluster and also the cluster with the highest biomass yield. Figure 6 shows the dendrogram view of the genotypes after the cluster analysis. Clusters 2 and 5 and 3 were genotypes with appreciable yields, which matures within the early maturity bracket and could fit into the Sudan savanna growing season. Conclusion: short duration cowpea has become a priority in the dry savannas due to the ever-shortening rainy season. Genotypes such as Mars16, Mars2, Supermagic 78, among others, were identified as early maturing high-yielding genotypes with plant and pot architecture that cowpea growers within the region prefer. Multi-location evaluation of these promising lines is needed to confirm their performance. We would like to acknowledge the support of our donors and our collaborators, especially from the Kwame Nkrumah University of Science and Technology, CSIR SARI, University of California, Riverside. Thank you for your attention.